Good morning, and I'd like to welcome everybody to today's webinar on frozen sample aliquotting. Today we'll be showing you how we can revolutionize your lab workflow with the handheld tissue extractor tool. So thank you to everyone for joining us today. I'm Jaquan Geathers, the Marketing Associate for MBA Scientific. Before we get started, I wanted to introduce our co-host, Kathy Rush, sorry about that, the Director of Science and Quality from Basque Engineering. And I just want to let everyone know really quickly before we get started that this webinar is being recorded. So if you need to leave at any point, don't worry at all. The recording will be available for everyone by at least the end of the week or the end of next week. And your microphones and cameras will be muted and turned off as well. So if you would like to ask a question, just drop your questions in the chat. And if you have any specific inquiries or prefer a private discussion, you can email us after the webinar as well. I would like to, quickly reintroduce ourselves. As you may know, we're MBS Scientific, the distributor of laboratory products serving the life science industry across 10 countries. We provide an extensive range of products, including but not limited to sample collection kits, sample stability reagents, storage tubes, barcode readers, rack thawing stations, tube labelers, laser markers, and more. In short, MBS Scientific is your trusted partner for just about all your laboratory needs. Okay, so now it's time to dive into the heart of the presentation. If Kathy Rush would like to take the stage and discuss frozen aliquotting and demonstrate the revamped handheld tissue extractor. So without any further ado, Kathy, the floor is yours.
Thank you, Jaquan. Thank you, everyone, for joining us this morning to learn a little bit more about frozen aliquotting and Basque Engineering's newly released tissue extractor, which is a handheld tool. Today's demonstration, I hope, will allow you to see what we can do to help with your frozen samples and help with your workflow. So going forward, I'm going to kind of review frozen sample aliquotting because thawing is not cool. Next slide. MBS and Basque Engineering Science are partners in frozen sample solutions for stable, reliable, and reproducible results. Next slide, please. Okay, so why is frozen sample aliquotting so important? Well, there's 7 concepts. 1, it improves sample integrity and preservation. It eliminates sample contamination. It increases sample utilization, allowing for reaccess. It works with your samples safely. It delivers sample consistency. It ensures reliable and reproducible data. And it's time efficiency for scientists. Next slide, please. Okay, so the first thing that we're going to talk about is it improves sample integrity and preservation. How does it do that? So it eliminates the freeze-thaw cycle that can degrade analytes, causing biased data in downstream analyses. It also stabilizes those labile molecules. Next slide. Eliminates sample contamination. Our patented technology uses nuclease-free coring probes to extract out aliquots, cores, from frozen biotissues feces. Both the parent and the aliquot samples are kept frozen through the entire process, ensuring preserved, high-quality samples for use in downstream assays. Next slide, please. Increases sample utilization and reaccessibility. 1 or multiple specific size cores are extracted out, leaving behind an intact parent sample. Parent sample is returned to the -80 freezer with reaccess later on, free of contamination. Next slide. Working with samples safely. It eliminates manual slicing and cutting of tissue, and it limits handling of raw feces. Next slide. It delivers sample consistency. It By generating, it distributes small, uniform, specific volume aliquots in a controlled and efficient manner. Next slide, please. Aliquots thaw faster than a full sample, improving sample consistency and reproducibility of data for results. Samples kept frozen retain their original analyte value because they are not degraded due to the freeze-thaw cycle. It also is a time savings for improved workflow. Next slide. Who benefits from a handheld tissue extractor? So research laboratories who are working with less than 50 samples per week. Frozen sample types that the tissue extractor works well with is tissues and feces only. The largest aliquot size attainable is about 50 mgs. Next slide. All right, so now what we're going to do is I'm going to show you what the kit entails. So the kit entails what we call a probe nest. The nest holds up to 6 probes. A probe looks like this. And what it is at the bottom are serrated edges. And this is what drills into your frozen sample. These are nuclease-free probes. The kit comes with a box of probes. There's 250 probes in here. There's 10 bags, each carrying 25 probes. These probes have been assembled, washed clean in a clean room, and then packaged into RNase, DNase, nuclease-free bags, and then put into the box. The kit also comes with the handheld tool, and this is what it looks like. It's very lightweight. It's made out of PEEK. It is autoclavable, so no worries there, or it can be washed down with a bleach wipe. Okay. So now what we're going to do is I'm going to go through the process of the actual handheld tool. Next slide, please. All right. So as you can see, the way that we do it is you're going to fill up your ice bucket, your dry ice bucket, with dry ice. And then what you're going to do when you take your nest is you're going to fill the nest up with 6 probes. Once you have 6 probes put in, you put it into the dry ice bucket to chill for about 15 minutes. You're going to use your processing tray or whatever you want to use to go ahead and put your sample on. The kits are optional, that if you want to have a tray, that can be purchased as an extra. Okay, so we're going to pretend that it's been 15 minutes, our probes are chilled, our processing tray is chilled, and now we're ready for our sample. So next slide, please. All right, so the tool has on the top button a lock and an unlock. And as you can see on the T-bar of the T is an etched indentation. To start the process, you're going to turn the tab into the lock position that lines up with the etched tab, so that the button, if you push down on it, does not move. Next slide. Now, in order to load a probe, you're going to take 2 fingers around the bottom edge of what I call the crown of the barrel, and you're going to push up. And when you push up, you will expose the inside of the barrel. The inside of that barrel is the same shape as my probe's hub. So what you're going to do is you're going to insert the probe and then release your 2 fingers so that the barrel covers now the probe, and it is now locked in place. So now what you're going to do is you're going to process your sample. I'm going to get my sample. So I'm going to put my sample on my processing tray, which is on the dry ice, which is now chilled. I'm going to take my sample, I'm going to take my probe, and I am going to press down into the sample, and at the same time, I'm going to turn the sample, giving it an even pressure. When it is done, I'm going to release the button. I'm going to turn it to the unlock position, and then you are going to press out your core. Oh, sorry about that. Oops, I just lost it. Hold on again. I don't want to lose it again. But there you can see the core. I better put it down before I lose it again. Now, here's your core. That's about a 40 to 50 meg core. And at that point, you have 2 options. You can take your tweezers and put it into your cryo tube that has been sitting on the dry ice chilled, or you can actually push the core into the tube. And then you just push up and release the probe and do it again. So I'll do it again. I'm going to lock, push in, release, and now I'm going to core. Going to unlock and press into my tube. And release. It's that simple. And that's it. Next slide. I'll take questions. Are there any questions?
So I'm not really seeing any questions in the Q&A in the chat. However, someone did leave a question when they registered, and that was, how can this system ease the tissue chipping process?
So that's a great question. So most people that are working with tissue are using knives and scalpels. And we know for a fact through other customers that when you are cutting or chipping your tissue, you can cut your fingers. There's no chipping, there's no cutting. The other thing that we realize is that when people do the cutting, they don't get a nice piece of tissue of the specific size they need to run their assays. It's very hard to take a chunk of tissue and realize, well, I need 50 megs of that, or I need 40 megs of that. It's very hard to quantify the amount of tissue that you have, number one. And number two, you want to make sure that you are not contaminating the actual tissue. When the tissue is in a container or even on the slab of the tray, there is no cutting. You are actually putting in a tool and I am really not cutting. I am not chipping. I am literally going in to the tissue. The only thing I'm doing is I'm holding the tissue tube. I come out and now I'm expelling a very nice round cylindrical core that can be used for processing. And then I release and I get rid of the probe. So here are the two cores that I was able to take. If you take a look, they're the same size. They're between 40 and 50 megs. They're cylindrical. I just did two cores for you. Now I still have my tissue. I didn't use the same probe. I used a different probe, so there's no contamination. And now I can put my sample, my parent sample, back in the freezer because I didn't thaw it. And now I have two really nice aliquots to run my assay on. No cutting, no chipping. I got the same size core 2 tries in a row, except for the one that I lost. Any more questions?
Yeah, someone asked, can the size be adjusted?
The size will really, so there's no measurement on the tool where you can plug in 10 millimeters or 10 megs or 15 megs or 20 megs. It really is dependent upon your strength and the pressure that you are going to put on in order to get into the tissue. It will not do any more than 50. So you are limited to 50, but if you need 100, you take 2 cores. If you want to take less than that, then you just don't press that hard into the tissue. Great question. Next.
Okay, I see someone maybe asking a question. Yeah, 2 questions. The first one that popped up was, what kind of tissues can be used?
Great question. So just about any tissue that you are working with will cut into using our probes. So whether you're working with heart, muscle, liver, whether it's animal or human, and you're trying to do an assay on a very small piece of tissue, this is ideal. So we have customers that have very small amount of tissues that they're working with, and they want to preserve as much of that tissue as possible for later use. So they use my extractor to go into those tissues, get out exactly what they need for their assay without wasting the entire tissue, because obviously the tissues are very precious and valuable, so they want to at least try to minimize the amount that they use and need, and this extractor is able to allow them to do that. It works great on feces too. Next, I think there was a second question.
Yeah. The next question was, what's the advantage for the tissue extractor compared to the disposable tissue puncher?
That's a great question. And I think with my tool, you get a nice cylindrical aliquot which can thaw very easily. I think with the tissue puncher, I have seen it. I don't think you get the consistency of the small, nice cylindrical core. And definitely with the puncher, you're going to get the same size every single time. Whereas with the tissue extractor, you can vary what you need based on the pressure that you put on the tool coring into the piece of tissue or feces. Great question.
Okay, so before we move on, are there any more questions? Give everyone a little bit of time to ask. Okay, I don't see anybody typing any questions, so I think that wraps that part up.
Great.
If you do end up having questions in the future about this, feel free to reach out to us. Our website is listed right there, nbsscientific.com. You can also email us at info@nbsscientific.com as well. Or if you prefer to call, our phone number is listed there as well. This also, as again, this is recorded, so you can always come back and check this information out and rewatch the webinar if you'd like. But other than that, I'd like to thank everybody for joining us today for this webinar. I'd like to thank Kathy again for presenting the tissue extractor. She did a great job. So if there is, again, any questions, don't hesitate to reach out to us. And I think this pretty much concludes our webinar today.